scientists have created living drugs that hunt cancer for over 10 years from a single injection. These aren't traditional medications that get processed and eliminated by your body. These are genetically modified versions of your own immune cells that patrol your bloodstream indefinitely, seeking and destroying cancer wherever it tries to hide. The University of Pennsylvania has patients who remain cancer-free more than a decade after treatment, with engineered T cells still circulating and ready to attack any returning malignancy. Welcome to Curiosity AI Podcast, Episode 20. Today we're exploring CAR T cell therapy, a breakthrough that fundamentally challenges how we think about cancer treatment and pharmaceutical development. Traditional chemotherapy destroys both healthy and cancerous tissue indiscriminately. Radiation therapy damages surrounding organs. Surgery removes accessible tumors but cannot address microscopic spread. CAR T therapy represents something entirely different, transforming your immune system into a personalized cancer-fighting army. The process extracts T cells from patients, genetically engineers them to recognize specific cancer markers, then reinfuses them as microscopic assassins. These modified cells multiply in your bloodstream and form memory populations that remember cancer signatures permanently. We're not discussing theoretical treatments or early-stage research. 6 CAR T therapies have FDA approval, with over 30,000 patients treated worldwide. The regulatory landscape shifted dramatically in 2025 when the FDA eliminated major access barriers while maintaining safety monitoring for rare long-term risks. The economic implications are staggering. Single treatments costing $400,000 to $500,000 can potentially eliminate lifelong cancer management expenses. But the manufacturing complexity and specialized delivery requirements create access challenges that extend far beyond pricing. Today, we'll examine the science behind living drugs, analyze clinical outcomes that span over a decade, and assess the realistic timeline for expanding these approaches beyond blood cancers into solid tumors and autoimmune diseases. CAR T therapy begins with leukapheresis, where patients' blood passes through machines that extract T cells while returning other blood components. These harvested cells undergo genetic modification in specialized laboratories using viral vectors that insert synthetic receptor genes. The chimeric antigen receptor itself represents sophisticated bioengineering. Unlike natural T cell receptors that require complex activation signals, CARs provide direct recognition of tumor antigens combined with built-in activation domains. The most common target is CD19, a protein expressed on B-cell malignancies including acute lymphoblastic leukemia and large B-cell lymphomas. The genetic modification process typically requires 2 to 3 weeks, though recent advances have achieved manufacturing in under 14 days. During this period, patients often receive bridging chemotherapy to prevent disease progression. The modified cells are expanded to therapeutic doses, usually millions to billions of cells per infusion. Quality control measures ensure product safety and potency. Each batch undergoes sterility testing, genetic stability assessments, and cellular viability measurements. The manufacturing complexity explains both the high costs and the limited number of qualified production facilities globally. Once reinfused, CAR T cells encounter their target antigens and activate rapidly. They release cytotoxic compounds that destroy cancer cells while proliferating to match tumor burden. Most importantly, some differentiate into memory cells that provide long-term surveillance against cancer recurrence. This memory formation distinguishes CAR T therapy from traditional treatments. While chemotherapy effects diminish over time, engineered T cells can remain functional for decades. The University of Pennsylvania's longest-followed patients show detectable CAR T cells in blood and bone marrow over 10 years post-treatment. The clinical evidence for CAR T efficacy spans multiple cancer types, with the most compelling results in hematologic malignancies. In pediatric acute lymphoblastic leukemia, complete remission rates reach 90% in heavily pretreated patients who had exhausted other options. The ZUMA-1 trial established axicabtagene ciloleucel as standard care for relapsed refractory large B-cell lymphoma. 5-year follow-up data show approximately 40% of patients remain in complete remission without any additional treatment. These represent genuine long-term survivors in diseases that were universally fatal before CAR-T therapy. Multiple myeloma applications demonstrate similar durability. Ciltacabtagene autoleucel shows that roughly 1/3 of patients remain alive and progression-free 5 years after treatment. The FDA's approval for earlier treatment lines reflects growing confidence in favorable benefit-risk profiles. Real-world evidence studies validate clinical trial results while revealing important limitations. Response rates are slightly lower in heterogeneous patient populations compared to carefully selected trial participants. Elderly patients and those with significant comorbidities show reduced efficacy, though substantial numbers still achieve durable remissions. Treatment failures typically result from 3 mechanisms. Antigen escape occurs when cancer cells lose the target protein, rendering CAR-T cells ineffective. T-cell exhaustion develops when engineered cells become dysfunctional over time. Immunosuppressive tumor microenvironments can inactivate CAR-T cells before they eliminate all cancer. Understanding these resistance patterns has informed next-generation CAR designs. Dual antigen targeting reduces escape potential. Enhanced co-stimulatory domains combat T-cell exhaustion. Combination approaches with checkpoint inhibitors or other immunotherapies address microenvironment suppression. The quality of life implications extend beyond survival statistics. CAR-T responders often return to normal activities without ongoing treatment requirements. This functional cure concept represents a paradigm shift from managing cancer as a chronic disease to potentially eliminating it through single interventions. The regulatory landscape has evolved rapidly as experience with CAR-T safety management has accumulated. The FDA's June 2025 elimination of risk evaluation and mitigation strategy requirements represents a watershed moment for patient access. REMS programs previously mandated extensive administrative procedures, including healthcare provider training and restricted distribution systems. While important for early safety monitoring, these requirements created significant barriers for treatment centers and delayed patient access to potentially life-saving therapy. The elimination reflects confidence gained from treating over 30,000 patients worldwide with manageable safety profiles. However, safety vigilance remains paramount through continued post-market surveillance and updated prescribing information. The most significant safety concern involves secondary T-cell malignancies identified through post-market surveillance. The FDA's April 2024 boxed warning quantifies this risk, though European Medicine Agency data suggests approximately 38 cases among 42,500 treated patients, representing very low absolute incidence. These secondary cancers appear to result from viral vector integration near oncogenes or tumor suppressor genes during the genetic modification process. While the risk remains minimal compared to benefits in life-threatening cancers, long-term monitoring protocols now extend to 15 years post-treatment. The regulatory streamlining has practical implications for healthcare delivery. Treatment center certification requirements have been simplified, potentially expanding qualified facility networks. Patient requirements to remain near treatment centers have been reduced from 4 weeks to 2 weeks, significantly reducing logistical burdens. International regulatory harmonization efforts progress slowly, with the European Medicines Agency and Japan's regulatory authority adopting similar but not identical approval pathways. These differences create complexities for global development, while reflecting varying healthcare system priorities. Manufacturing represents both the greatest limitation and most active innovation area for CAR-T therapy. The complexity of producing personalized cellular therapeutics at scale requires entirely new approaches to pharmaceutical manufacturing. Traditional pharmaceutical production involves chemical synthesis with predictable inputs and outputs. CAR-T manufacturing begins with variable patient starting material, requires live cell culture, and produces biological products with inherent batch-to-batch variability. Quality control measures must ensure safety and efficacy for each individual product. Vein-to-vein time remains a critical metric affecting both patient outcomes and access. Studies demonstrate that manufacturing delays beyond 30 days correlate with worse clinical outcomes, likely reflecting disease progression during treatment delays. Patients whose cancer advances while waiting often become ineligible or experience reduced efficacy. Recent technological advances have achieved remarkable improvements. Novartis has demonstrated manufacturing completion in 24 hours using automated platforms, though end-to-end delivery still requires quality testing and logistics. Point-of-care manufacturing systems enable local production at treatment centers, potentially reducing total timelines to under 1 week. Allogeneic or off-the-shelf approaches aim to eliminate patient-specific manufacturing entirely. These products use T cells from healthy donors, genetically modified to reduce graft versus host disease risk while maintaining anti-cancer activity. Early clinical results show encouraging response rates, though persistence duration remains shorter than autologous products. The most ambitious development involves in vivo CAR-T generation, using lipid nanoparticles to deliver genetic modifications directly to patients' T cells. This approach could theoretically reduce treatment to simple injections, eliminating manufacturing complexities. First-in-human trials began in 2024, but efficacy data remains very limited. Non-viral gene editing approaches using CRISPR technology could address safety concerns about viral vector integration while enabling more precise genetic modifications. These methods potentially reduce secondary malignancy risks that prompted regulatory warnings. The expansion of CAR-T therapy beyond cancer represents one of the most intriguing developments in cellular therapeutics. Autoimmune disease applications demonstrate how targeted immune system modifications can address diverse pathological processes. Systemic lupus erythematosus patients treated with CD19-targeted CAR-T therapy have achieved drug-free remissions lasting over a year. The mechanistic rationale involves eliminating autoreactive B cells that drive autoimmune pathology, essentially resetting dysfunctional immune responses. Early results in myasthenia gravis show substantial clinical improvement with patients discontinuing immunosuppressive medications while maintaining disease remission. Multiple sclerosis applications demonstrate rapid inflammatory quiescence and MRI stabilization, though long-term follow-up remains limited. The success in autoimmune diseases has attracted significant pharmaceutical investment and clinical development activity. Multiple companies have initiated programs targeting various conditions, recognizing potential to transform treatment paradigms for chronic diseases affecting millions globally. Solid tumor applications remain challenging despite extensive research investment. The immunosuppressive tumor microenvironment inactivates CAR-T cells, while antigen heterogeneity enables cancer escape. Additionally, solid tumors often lack uniform target expression, complicating therapeutic targeting. Despite these challenges, encouraging results have emerged in specific contexts. Glioblastoma trials using dual antigen targeting have shown tumor shrinkage in approximately 2/3 of patients, though durability remains limited compared to hematologic applications. Combination strategies aim to overcome solid tumor limitations through complementary approaches. CAR-T cells combined with checkpoint inhibitors, bispecific antibodies, or tumor microenvironment modifying agents show promise in early trials by addressing multiple escape mechanisms simultaneously. The economic implications of CAR-T therapy extend far beyond headline-grabbing list prices. Comprehensive analyses must consider total cost of care over patient lifetimes, including avoided expenses from traditional treatments and quality-adjusted life years gained. List prices ranging from $400,000 to $500,000 reflect value-based pricing rather than manufacturing costs. While actual production expenses remain proprietary, industry estimates suggest $20,000 to $50,000 per dose, with pricing premiums reflecting research investments and curative potential. Cost-effectiveness studies consistently demonstrate favorable outcomes for appropriate patient populations. The Institute for Clinical and Economic Review found CAR-T therapy achieves approximately $68,000 per quality-adjusted life year, well within conventional cost-effectiveness thresholds for life-saving treatments. These analyses become compelling when considering alternative costs of managing relapsed, refractory cancers. Traditional salvage therapies often require multiple treatment cycles, prolonged hospitalizations, and supportive care for toxicities. Cumulative costs can exceed CAR-T expenses while providing inferior outcomes. Healthcare system capacity constraints present ongoing challenges despite manufacturing improvements. Treatment requires specialized facilities with intensive care capabilities, experienced nursing staff, and 24-hour physician coverage for managing acute toxicities. Geographic access remains limited with rural populations facing significant barriers. Insurance coverage patterns generally favor CAR-T therapy for approved indications, though prior authorization requirements often delay treatment initiation. The regulatory streamlining should facilitate coverage decisions by reducing administrative complexity for payers and providers. Real-world outcome registries reveal concerning disparities in access based on race, geography, and socioeconomic status. These databases highlight ongoing equity challenges in precision medicine delivery that extend beyond individual treatment costs to systemic healthcare access issues. The trajectory of CAR-T therapy points toward increasingly sophisticated and accessible treatments, though realistic timelines require careful assessment of remaining challenges and ongoing development priorities. Next-generation CAR designs incorporate safety switches, enhanced trafficking capabilities, and resistance to tumor microenvironment suppression. These improvements aim to expand efficacy while reducing toxicity risks, particularly important for applications in non-fatal conditions like autoimmune diseases. Universal platforms using allogeneic donors could democratize access by eliminating patient-specific manufacturing constraints. Current approaches show promise, but require overcoming persistence limitations and optimizing immunosuppression protocols to prevent rejection while maintaining efficacy. Artificial intelligence integration promises optimization across multiple therapy aspects, from patient selection to manufacturing process control. Machine learning algorithms could predict treatment responses, identify optimal CAR designs, and guide personalized dosing strategies for individual patients. The convergence of gene editing advances with cellular therapeutics enables more precise modifications while potentially reducing safety concerns. Base editing and prime editing technologies allow targeted genetic changes without double-strand breaks, potentially decreasing unintended consequences. However, several critical challenges require resolution before CAR-T therapy achieves its full potential. Solid tumor applications need breakthrough approaches to overcome microenvironment suppression and antigen heterogeneity. Manufacturing scalability must improve to serve global patient populations. Long-term safety profiles require continued monitoring as treatment numbers expand. The expansion into autoimmune diseases, while promising, requires careful risk-benefit evaluation in conditions that are chronic rather than immediately life-threatening. The immune suppression necessary for successful treatment could create infection vulnerabilities or other complications. CAR T-cell therapy represents one of medicine's most remarkable achievements, transforming theoretical concepts about harnessing the immune system into clinical treatments that offer genuine hope for cancer cures. The ability to engineer personalized cellular therapeutics from patients' own immune systems exemplifies precision medicine at its most sophisticated level. The documentation of complete remissions lasting over a decade establishes proof of principle for living drug approaches. The regulatory evolution from restricted experimental therapy to streamlined clinical treatment reflects growing confidence in safety management and clinical benefit. The elimination of administrative barriers should expand access while maintaining appropriate safety vigilance. Manufacturing innovations continue progressing toward more efficient and accessible production methods. While current timelines and costs remain challenging, the trajectory toward point-of-care manufacturing and potentially in vivo generation offers hope for democratized access. The expansion beyond cancer into autoimmune diseases opens entirely new therapeutic possibilities. The mechanistic success in resetting dysfunctional immune responses suggests applications across numerous chronic inflammatory conditions affecting millions of patients globally. However, critical limitations require honest acknowledgement. Success remains concentrated in blood cancers, with solid tumor applications showing modest progress despite intensive research. The high costs and manufacturing complexity create equity concerns that extend beyond individual access to systemic healthcare delivery challenges. The living drug concept fundamentally challenges traditional pharmaceutical models based on chronic treatment approaches. The potential for single interventions to provide decades of protection represents a paradigm shift with profound implications for how we approach disease treatment and healthcare economics. For patients facing blood cancers that previously carried universal fatality, CAR-T therapy offers transformational hope. As technology advances and access expands, these benefits should reach broader patient populations while extending into new disease applications. The success of CAR-T validates investment in cellular and gene therapy platforms that are being adapted for multiple medical applications beyond oncology. The convergence of genetic engineering, artificial intelligence, and cellular therapeutics promises even more sophisticated future treatments. Thanks for joining us on episode 20 of Curiosity AI Podcast. If this exploration of living drug technology and personalized cellular therapeutics engaged your curiosity, subscribe for continued analysis of scientific breakthroughs reshaping medicine and human health. Until next time, remember that the future of medicine is being engineered at the cellular level, transforming how we combat disease from within our own biological systems.